Hello everyone, today we're going to dive into the world of gene mapping using a classic example, the 3-point test cross in the garden pea plant. We'll be working through a problem to determine the three main things, the linkage between genes, the map distance that separate them on a chromosome, and a fascinating phenomenon called interference. Let's start by setting up the problem. You are looking at the tree in pea plant. Pod color is green is dominant over yellow, seeding color is green is dominant over bluish green, and growth type, normal growth is dominant over the creeper type. We start with two true breeding parents. A normal plant with all dominant traits GG, HH, and NN is crossed with a creeper plant with all the recessive traits GG, HH, and NN. This produces an F1 generation that is heterozygous for all the three genes GG, HH, and NN. And importantly, because the dominant allele all come from the same parent, they are on the same chromosome in what we call decoupling or cis configuration. The most important rule in all 3-point test cross is this: the two largest classes are the parental and the two smallest are the double crossover or DCO. Here, we see the parental classes are by far the most common with about 4,100 offspring. They are the non-recombinant chromosomes that were passed on from the F1 parent. The double crossover classes are the rarest with all 26 offspring. This requires two crossover events to occur simultaneously, which is very unlikely. The classes is between our single crossover SCO in region 1 and region 2. Identifying these extreme classes is our first and most critical part.
For part A, we need to find how a three genes are linked in the trihybrid parent. First, you solve your AF2 classes by frequency. The two largest numbers, 2059 and 2041, are parental, not crossover type. And the two smallest, 15 and 11, are double crossover. Now compare a parental class to its matching double crossover class. Parental give green part, green signet normal. The double crossover give green part, black signets and creeper. Notice the part color staying the same, which is green in the both. Only signet color and creeper tray change together. In a double crossover, the middle gene is the one that flip while the two outer genes stay linked. Since part color didn't flip, part color is the middle, so the gene order is signet color, part color and creeper.
Alright, so for question B, we need to calculate the map distance between our genes. The formula we always use is recombinant divided by total offspring times 100, and that gives us the distance in centimorgan. Let's start with region 1 first, which is the distance between gen G and gen H. To get our recombinant here, we have to add two things: the single crossover that happened in region 1, which is the 308, and the double crossover, which is 26. We include the double crossover because remember, a double crossover event also have a crossover between G and H. So 308 plus 26 gives us 334 recombinant. Now, we take the 334 and divide it by the total offspring, which is 4,997. That gives us about 0.0668. Then, we multiply that by 100 and we get 6.68 centimorgan. So, the distance between G and H is 6.68 centimorgan. Now for region 2, which is the distance between gen H and gen N, we do the exact same thing. We add the single crossover in region 2, which is 563, to the double crossover, which is 26, giving us 589 recombinant. Then we divide 589 by 4,997, which gives us about 0.1179. Multiply that by 100, and we get 11.79 centimorgan. So the distance between H and N is 11.79 centimorgan. And that's our answer for question B. The genetic map look like this: G, then 6.68 centimorgans away is H, then 11.79 centimorgan away is N.
First, we identify the parent, single crossover and double crossover. From the data, the observed DCO is 15 plus 11, which is equal 26. The expert DCO using the recombination frequency between genes is 0.1179 multiplied by 0.0668 multiplied by 14,997, giving value of 39.35. Then COC equal 26 divided by 39.35, which is 0.6607. Finally, interference is calculated using formula. Interference equal 1 minus COC. So 1 minus 0.6607 give 0.3394.
To calculate our expected double crossing over event, we first compare our parental and DCO class, which provide the port color gen sit in the middle. Next, we calculated the individual recombination frequency for both regions, assuming the independent adsorbent, meaning the crossover don't interface with each other. We multiply those two frequencies together. This gives us an expected DCO frequency of 0.79%, which translates to 39.37 plant total. This expectation is split equally between our two reciprocal phenotype category.